Roughly 1 in 12 men, but only about 1 in 200 women, experience some form of color vision deficiency, a striking sex difference that traces directly back to the specific genetics of how color vision works. Understanding why color blindness affects men so much more often than women reveals fundamental facts about both the biology of color perception and the mechanics of X-linked genetic inheritance. The trichromatic theory of color vision, supported by direct physiological evidence, holds that human color perception depends on three distinct types of cone photoreceptor cells in the retina, each maximally sensitive to a different range of light wavelengths, roughly corresponding to short, medium, and long wavelengths, commonly though imprecisely labeled blue, green, and red cones. The brain determines perceived color by comparing the relative activation levels across all three cone types simultaneously, allowing the visual system to discriminate millions of distinct color shades from just three types of receptor input. The specific genes encoding the light-sensitive photopigment proteins in medium wavelength and long wavelength cones are both located on the X chromosome, meaning that since men possess only one X chromosome, a single defective gene copy is sufficient to produce color vision deficiency, while women, possessing two X chromosomes, would need a defective gene copy on both X chromosomes to show the same deficiency, a considerably less probable genetic event, explaining the dramatic sex difference in color blindness prevalence. Red-green color vision deficiency, by far the most common form of color blindness, results from missing, reduced, or spectrally shifted function in either the medium wavelength or long wavelength cone photopigments, making it difficult or impossible for affected individuals to reliably distinguish between certain shades of red and green that appear clearly distinct to someone with typical trichromatic color vision. Protanopia involves complete absence of long wavelength cone function, while deuteranopia involves complete absence of medium wavelength cone function, with anomalous, partial versions of each also occurring. Achromatopsia, complete color blindness in which the affected individual perceives the entire visual world only in shades of gray, is an extremely rare condition, affecting far fewer than 1 in 10,000 people, resulting from a near-total absence of functioning cone photoreceptors of any type, forcing vision to rely entirely on rod photoreceptors, which are highly sensitive to light but incapable of distinguishing color, and are also poorly suited to detailed vision, meaning achromatopsia. Typically involves significantly reduced visual acuity and extreme light sensitivity alongside the complete absence of color perception. Color vision deficiency creates genuine practical challenges across numerous everyday and occupational contexts that assume typical trichromatic color perception, including certain color-coded electrical wiring and circuit diagrams, some traffic signal configurations, color-coded maps and data visualizations, and specific career paths, including some aviation, electrical, and military roles, which maintain color vision screening requirements given the safety-critical importance of accurately distinguishing specific color-coded signals in those particular professional contexts. Several assistive technologies have been developed to help individuals with color vision deficiency better navigate color-dependent tasks, including specialized tinted glasses that filter specific wavelengths to enhance the perceived contrast between commonly confused colors for some forms of red-green deficiency, and smartphone camera applications that use image processing algorithms to identify and label colors or to remap difficult-to-distinguish color combinations into a range the user can more easily perceive, though these approaches vary considerably in effectiveness across different individuals and specific types of color vision deficiency. Color vision deficiency offers a striking, real-world illustration of trichromatic theory in action. The specific genetic location of cone photopigment genes on the X chromosome directly explains both why color blindness exists and why it affects men so much more frequently than women, connecting basic sensory physiology directly to genetics and everyday practical experience.